All right, here it is. Here are our three tests. I don't know if that's the order they actually end up in this presentation, but let's say at the outset you need to know what their name is, what they test for, how to actually perform the experiment, including what reagents go into which mixture and how much of each unknown goes into each mixture. And then another thing you need to know is what the results actually look like. So you have to be able to see a picture of one of these tests run and say, oh, that's a positive reaction or that's a negative reaction. And then most of the questions are actually going to be about interpretation, such as you see a positive test for Benedicts but a negative test for iodine. What does that tell you about the thing you're looking at? And there is an answer to that. Hopefully think about it, and I'll leave a note at the end of this presentation when we're done with it. So here they are. Benedict's test looks like this. You start with a nice clear solution, but blue. That's because of the copper ions that are in there. When you mix this with any monosaccharide and some disaccharides, okay, we are going to test you on this. Write it down. Any monosaccharide or some disaccharide. That means a simple sugar or an only slightly complex sugar, a dimer, two simple sugars linked together. And it's only some of those disaccharides that will react. I think that they are listed in your notebook, so check that out. So again, we start with a copper solution. Copper reacts with the aldehyde group in the simple sugar or the exposed aldehyde group that's in a subset of disaccharides and leads to this orange precipitate and a change from copper that results from a reduction of copper 2 to copper 1. Okay? So you may remember blue solutions being copper from chemistry class. It's still the same copper, it's still the same solution, and when you change its oxidation state, it gets actually different physical properties. So this is a positive result here on the left. Okay. So remember we talked about what the chemical groups were that were biologically relevant. So look in your lab notebook and pay particular attention to what an aldehyde group looks like. It's a double bond oxygen to a carbon next to a hydrogen. It has such and such reactivity that makes this reaction possible. If you really want to get into the gory details, I'm sure they'll be happy to cover it for you in chemistry class. I don't think we're going to test you on it, though. I can also tell you, because I was a chemistry major, I'm going to leave it out now, though. But this is the reaction. You should probably be able to reproduce this schematic here. Notice, okay, some of the general properties of a sugar, why this works. So, any monosaccharide should have an aldehyde group in it. Note that a sugar is a general thing. It's anything that basically has this carbohydrate formula, 1 carbon for every 2 hydrogens for every 1 oxygen. In this class, we are pretty much going to talk about only the 6-string ones. Okay? There's 3, there's 4, there's 5. The 5 ones are important, like ribose for RNA and such, but still, we're going to stick with the 6 carbons. So I'd say which of these is the aldose? Which can you point to and say that there's an aldehyde group? Class, on the count of 3, left or right? 1, 2, 3. On the right, you can see here's a hydrogen next to a double bonded carbon with oxygen. Okay. That's more in depth than I want to get, so let's move on. That's the cyclic form. Okay, test number 2, the iodine test. We discussed this earlier, but remember that coiled complex carbohydrates such as glycogen or starch will take iodine out of solution, iodine that's in solution as an ion, so potassium iodide, and it will remove it as iodine 2 by wrapping all those complex carbohydrate arms present in this one molecule of starch or glycogen around the iodine and just dropping this precipitate. So when you have a positive result, it looks like this blue stuff here on the right. Going in, your solution should be whatever your sample is, plus this kind of little bit of a brown color from the actual detection reagent. Okay. Notably, this test will not detect simple carbohydrates because there's no long arm of carbohydrates to wrap around the iodine. This is what it looks like. Yes. So you see plenty of sugars wrapped around these. Why don't you freak out? That'll be great. Here we go. Wrapped around these iodine 2 molecules. Remember, iodine's a really big atom, so this is actually to scale. But you need a long row of sugars to get this result. So it's only starch and glycogen. We don't deal with. This is actually a note to a previous class, but I still like to make the distinction. Glycogen is present in animals. It has a slightly different branching pattern than starch. And starch is in plants, and it still has different pattern than the glycogen. I tend to use the words interchangeably. Some others would as well. Now, we got some leftovers here. I will probably delete this, but this is what glycogen looks like, branched out. It's a huge molecule, beads on a string. I'll contrast that with cellulose. I'm sorry, this is out of order, but notice that this is a flat sheet. That's why wood is not terribly water soluble. Finally, the Burette. I say it Biuret. I do not know the proper pronunciation of this test, but this is the one that's different from the others because it does not detect carbohydrates. It detects protein. So if the two previous tests parse what kind of carbohydrate you're looking at. Is it simple or is it complex? This one only tells you if there's protein or not. I actually am an aficionado of stereograms, but I'll suffice it to say, let me know if you can see this or if you want to see it, and it becomes 3D if you merge it with crossing your eyes. But here are the major points of this test. It has to be in alkaline solution. What's alkaline solution? Basic. What's a base? In this case, sodium hydroxide. That's what we're going to be using for this class. Okay. Why do we need to add base to this solution? There are actually two reasons. One falls into the mystical, nobody really understands it category, which is almost perfectly true strictly because it deals with quantum mechanics. And the other is we need to denature the protein to get access to the peptide bonds. I guarantee this question will be on the test. Why do you need to add base to this solution? You need to put enough base into solution such that the hydrogen bonds holding these molecules together in this shape are overpowered by the amount of negative ions present in the solution, and they just unfold. When they unfold, we get access to the peptide bonds, and hopefully that's shown in the next slide. This is what the Burette test looks like. So you will put into your solution, which way does it go? Have a solution, add a couple of drops, I believe, of the copper detection agent. A negative test is red, and a positive test is purple. And that's a result of the copper getting somewhere lost between copper 1 and copper 2 on a quantum mechanic level. So again, you need to know that this is for protein. You need to know what it looks like and what goes into the tube and in what ratios. Here's the figure I was looking for. So this is what the reaction actually looks like to form a peptide bond. So it's two amino acids coming together to link this, to replace this oxygen here with this nitrogen. And we're going to what's the word, class? I wish it was denature. Dehydrate. Here we go. We're going to lose a water in the process. Specifically, we're going to lose this oxygen, this hydrogen, and one of these hydrogens. Okay, so we've got a peptide bond. This is the generic formula. Here it is again. So here's the quantum mechanic reason. the hydroxyl molecule gets in here, starts pulling electrons around, and this mechanism is not well understood. That's why I'm still punting, but that helps bring this copper to this bond, and it helps to change the oxidation state of that copper. But this gets in here, everybody in the mix is sharing electrons, and so again, copper's stuck between copper 1 and copper 2, so it's not red, it's not blue, it's purple. It actually makes sense intuitively too, which is weird. But again, we need sodium hydroxide for the other purpose as well, and this is the one that you want to be writing down when there's a question. Sodium hydroxide denatures the protein such that there's not a whole big mess of other amino acids blocking access of the copper to this bond right here.